Hello friends, welcome back to my channel, and today in this video we are going to study about tumor necrosis factor family cytokines. So the cytokines of tumor necrosis factor, so by the name you can understand that it helps in the necrosis of or the degradation of or it helps to destroy the cells that became tumorigenic in nature or the tumorous cells. So TNF related cytokines, these are unusual. Why these are unusual? That mostly till now whatever cytokines we learned, these cytokines are the small molecules that are secreted by a cell in the surrounding environment, and the receptors are present on some nearby cells. So these are soluble molecules, small sized soluble molecules. But tumor necrosis factor cytokines, these are unusual because they remain firmly anchored into the cell membrane. They are not soluble in nature, they remain anchored with the cell membrane. But we will see later that the few members of this families are also soluble in nature. So generally, because these are anchored to the cell membrane, they have type 2 transmembrane protein. That means they have a short intracytoplasmic N terminal and a long extracellular C terminal. They can also act as soluble mediators. Now, tumor necrosis factor beta is more commonly known as lymphotoxin alpha. Now, this is a soluble protein, whereas tumor necrosis factor alpha, it produced by activated macrophages and also other cell types like lymphocytes, fibroblasts, and keratinocytes in response to infections or inflammation. Lymphotoxin alpha can lead to increased expression of MHC glycoproteins. MHC means major histocompatibility complex. We learned that in T cells, we already learned that T cells can only recognize antigen when presented via MHC. Okay, so these are the presenters of the antigens. These are present on antigen presenting cells, and these are presenters of the antigen to the T cells. Even all the nucleated cells of our body also contain this MHC on their surface. Now, lymphotoxin beta, which is a membrane bound cytokinin and is important for lymphocyte differentiation. Similarly, one more family member, that is CD40L, is a cytokine expressed on the surface of T cell, and it is required to signal for B cell differentiation. That means it is present on the surface of T cell but required for the differentiation of B cell. Similarly, Fas ligand or CD95L, these are responsible for the apoptosis. Apoptosis means programmed cell death. Whenever the requirement of any cell is finished, whenever the function of any cell is done or its lifespan is finished, that time the body will induce apoptosis to destroy that cell whose life cycle has been finished or whose function is done. Now, if you talk about the receptors of this family, so the members of the TNF receptor superfamily, these are defined by the presence of cysteine rich domains, which are called CRD in short, in the extracellular ligand binding domain. Now, because they are cysteine rich, they can form disulfide bond loops, and individual members of superfamily can contain from 1 to 6 CRD. Although most tumor necrosis factor receptors are type 1 membrane protein, a few family receptors are cleaved from the membrane to form soluble receptor variant. Now, this is the signaling of tumor necrosis factor superfamily. This is the signaling which is called FAS. As you can see here, Fas ligand signaling. This signaling happens when, for example, any infection occurred in our body, and in response to that infection, some T cells were produced as an adaptive immune response to kill that antigens. One, the antigen is completely cleared off from the body. Now the extra T cells which the body produced to kill those antigens, now antigens are not present because all the antigens are cleared off. But body has produced some extra T cells. Now this leftover T cells need to undergo the apoptosis because now their function is done. All the antigens are cleared. Now the extra T cells which are left over, that needs to be destroyed, otherwise it can harm our normal cells of the body also. So how these can be destroyed? By the process of apoptosis, which is also known as programmed cell death. For that, the T cell will start expressing this Fas, and the ligand for that Fas is known as Fas ligand. So the T cell will start expressing this. Now once this Fas ligand is bind to the Fas is associated with FAD. What is FAD? That is fast associated death domain containing protein. Now this FAD consists of two parts. One is death domain, another is death effector domain. Now with this death effector domain, procaspase 8 is bound. Now the procaspase 8 is an inactive form. It will undergo activation, and once it is active, it will activate procaspase 7 and procaspase 3. Once it activates those, now the activated caspase 3 and 7 will activate apoptotic enzyme which remains inactivated. So who will activate this apoptotic enzymes? Caspase 7 and caspase 3, which in turn was activated by caspase 8. Once this apoptotic enzymes get activated, it will lead to apoptosis, that is programmed cell death. So, and the combination of all these, that is FAS, FAD and these procaspase 8, this is known as DISC, that is death inducing signaling complex. Okay, because they are responsible for the death of that particular T cells which are now left over and needs to be destroyed or cleared off from our body. Now, the signaling through tumor necrosis factor R1 receptor. Now, this R1 receptor is present in all the vertebrate cells. In all the cells of the body, this is present. Now, because this is also a receptor for tumor necrosis factor, now you can see here two pathways normally run when TNF binds with TNF R1. Either it will go to the apoptosis pathway or it will go to the survival pathway. Now, TNF-R1 contains DD, that is intracytoplasmic death domain. To this, TRADD binds. With TRADD, RIP1 and TRAF2 binds. This complex gets dissociated from the cytoplasmic domain and get released in the cytoplasm. Once it released in the cytoplasm, it will bind to FAD and procaspase 8 and will activate procaspase 8 into caspase 8. Now this activated caspase 8 will lead to apoptosis. And in the another pathway where the survival needs to take place, this complex, which is common in both the cases, what is the difference? For the apoptosis, this complex get dissociated from this cytoplasmic DD domain and get released into cytoplasm, whereas in survival pathway they remain attached to the DD domain, and to it various other proteins get attached, which finally activate TAB1, TAB2 and TAC1. This TAC1 can further activate either the NF kappa beta pathway or the RAS MAP kinase cascade, leading to the survival of the particular cell. Now, this NF kappa beta will in turn activate C flip. Once the C flip gets activated, it will stop. This type of arrow is for the blockage. So this will block the activation of caspase 8. So if the activation of caspase 8 get blocked, it cannot lead to apoptosis. There are two pathways, one leading to apoptosis, one leading to survival. So which pathway will win? Whether apoptosis pathway will win or the survival pathway win, this decision depends upon the race. Both the pathway will start their race. Whosoever will finish the race first is the winner. If before C-clip formation, caspase 8 gets activated and leads to the activation of procaspase 3 and 7, then it will lead to apoptosis. If before the caspase 8 activation C-flip is formed, then it will lead to the survival pathway. Clear? So this is a race between two pathways, the apoptosis and the survival. Whosoever wins the race, that will be the outcome of that particular cell. Now, this small decision that why at one particular situation apoptosis pathway will win, why in another situation survival pathway will win, is not clear completely till now. But till now, only this much is clear, that there is a race between the two, and whosoever wins the race will lead to the outcome of that particular cell. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.